Good afternoon, everyone, and welcome to another webinar sponsored by the North American Vascular Biology Organization. I'm Minar Cuervo from the University of Illinois at Chicago and also a member of the NAVBO Education Committee. I will be moderating today's session. We are pleased to welcome our speaker, Stefania Nicoli from Yale University, who will present her work entitled microRNA-Dependent Regulation of Biomechanical Genes Establishes Tissue Stiffness Homeostasis. At this time, I'd also like to welcome Tristan Driscoll, also from Yale University. He will monitor today's questions. Questions will be handled in two ways. Throughout the presentation, you can type your questions into the question box in the control panel. These questions will be answered at the end of the presentation. So Dr. Driscoll will compile them throughout the presentation and then pose the questions to Dr. Nicoli at the end. At the end of the question and answer period, provided that there's time, attendees will be able to ask additional questions. If you have a question then, please raise your hand by clicking on the hand icon on the left side of your control panel, and then I will recognize you, your mic will be unmuted, and then you will be able to ask your question live. Before we get started, I wanted to go over some logistical aspects. Throughout this webinar, you're able to switch between the phone audio and the computer audio in case you're having troubles. You can see this information in the audio sections of your GoToWebinar control panel. If you're experiencing technical problems, please click the Help tab at the top of the control panel. Scroll to the bottom of the Help screen, and then you'll see the technical support phone number. This webinar is being recorded and archived on the NAVBO website for future use. Dr. Nicoli received her PhD from the University of Brescia School of Medicine in Italy and performed her postdoctoral studies at UMass Medical School. Currently, she holds an associate professorship at the Yale University School of Medicine. Her lab has published 9 primary research manuscripts elucidating the cellular and genetic rules that control neural and neurovascular behaviors in the developing brain and the principles of vascular morphogenesis in zebrafish. She has received multiple awards for her accomplishments in the field of vascular biology, and she was the first woman to receive NAVBO Springer Award for a junior investigator in 2016. Today's webinar will present research on the role of microRNAs in regulatory pathways in homeostatic tissue stiffness and tension over time after changes in mechanical stress. These findings are an important step towards understanding the initial pathological alterations resulting in fibrotic and related diseases. Welcome, Dr. Nicoli.
Thank you very much. Welcome to everybody, and I really thank you, the NAVBO Educational Committee, to give this the opportunity to virtually meet all of you and to show what is cooking in my lab. But first of all, I'd like to introduce my lab and for more detail on what we like to answer and what we do, please feel free to reach my lab website. And today, I would like to highlight 2 people that are the protagonists of the story that I will describe in a few minutes. One is Tristan, that he will answer also your questions, so you can actually see him and recognize him if you see at some NAVBO conference. And the second one is Alberto Maria Moro, which he's now a computational biology in the Sloan Kettering Cancer Institute in New York. Just a sneak peek, this is a story that will be online this Monday in Nature Cell Biology, so I'll give you what is, so to speak, an exclusive somehow. Although I also would like to talk a little bit, what would we like to do in the lab and how actually we arrive to answer and to ask the question of mechanical homeostasis. So overall, my lab is very interested in what I think powerful question. So how the regulation of a gene is actually able to make what we all love, so this beautiful cardiovascular system. And of course, to answer this question, the embryonic development seems extremely suitable for that. And through the years, especially through undergrad, graduate school, and postdoc, I really realized that this is never actually a linear progression. So one gene, one vascular phenotype. Indeed, the genome often to take into account what is the environmental parameter and therefore adapt to that and to build the cardiovascular system. And by environmental parameter, I mean the stochastic nature of the gene expression, again, transcription as well as translation. They are never precise in terms terms of how much protein output each cell type need for a phenotype. A second environmental parameter are, of course, genomic variance, which is so popular right now in our genomic area, and especially in human disease. And finally, of course, the effect of environmental parameter more physically, such as the diet, the oxygen temperature, and so forth. So in essence, if we, the genome take into account all these environmental parameters, we actually realize that the vascular phenotype is never a unique and reproducible, but it's a window of phenotype, which is also called phenotypic variability. So indeed, although we are common as far as building the vascular structure, we are never identical one to each other. And that's because the genome communicates with the environment. So I like this question, particularly because it's very now well studied in developmental biology, and particularly there are very few observations in this regard. For example, one publication was reporting that a microRNA knockout, miRNA, in Drosophila melanogaster, and I apologize for the typo for the melanogaster fans. But in this sense, this particular mutant have a number of scrutella, which is sensory organ in this animal, which indeed, when the microRNA is missing, is very variable through a population of isogenic individual, while the wild type is always end up to be about 4. And this is true also for another microRNA knockout, such as LED7, with the number of vulva C. elegans. You may be thinking right now, well, what exactly, why do we care about phenotype? They are mostly sensitive to genetic and environment. So I actually think this is one of the most challenging questions in our current research, because it is indeed a bit headache that medicine had to understand how the genetic insult result to a different phenotypic window in disease. Let me give you an example. This is a very typical where a mutation in sodium sodium channel, in this case, the patient doesn't really manifest a focal phenotype, but manifests what is a window of phenotype through our individual, again, with the same exact mutation. And this individual, they also further susceptible to environmental parameters. So in essence, by trying to decouple how the gene expression control a phenotype in relate to different environmental parameters, maybe very well elucidate how our disease manifestation is so different in different patients. Of course, how do we approach this problem? The logic will be to follow the few examples previously established in invertebrates, so by analyzing knockout for microRNA, although it is very unfortunate that the majority of the microRNA knockout, especially mammalian, although abundantly expressed, as you can see in this Holt report about the expression of many microRNAs through development, like in particular by integrated LAG-Z, they show that indeed microRNA are abundantly expressed, but the main goal, so the knockout of this mouse, have no phenotype. So it's very hard to therefore visualize and quantify trait because there is literally no difference between the wild type. So for this reason, we decided at the time to initiate what is a reverse genetic screening to capture phenotypic variation using microRNA knockout. And for doing that, we designed microRNA knockout in zebrafish. And I hope that you probably know already that the advantage of the model being transparent and somehow easy to visualize in different contexts, in different vascular branches. For you can literally quantify any type of trait. And by trait, I mean, again, a phenotypic behavior of an endothelial cell in this particular case. So with that, we essentially generate a library that's briefly schemarized right here, in which first we understood how many microRNA are expressed through the zebrafish vascular development. Again, using this transgenic line, the labeled vascular cells via KD-RGFP, we isolate the endothelial cell, and by differential expression, we understand which microRNA were predominantly rich. Then it took about 5 years, but we did it. We knock out every single microRNA in the genome that, again, for microRNA enriched in this vascular context. And we've been pretty successful in showing that these are true homozygous loss of function mutation. But again, spoiler alert, and none of these animals have, again, strong phenotype. So therefore, we decide to analyze much more deeply each single cell behavior and trait. And I want to give you only a taste of what we identify by showing you the phenotype and the analysis on one microRNA, miR-139 knockout. So this microRNA was founded as expresses in the intersomatic vessel of the trunk of the zebrafish embryo, and we were able to identify there was function in this vessel by using this microRNA biosensor, which will be very important also for my discussion later on. This is a bidirectional endothelial vascular promoter driving nCherry and GFP. The GFP do not have any microRNA TUTR binding site, but the cherry contained the miR-139 binding site. So in essence, if you inject this in an animal model, you can visualize a structure in which the microRNA is essentially active, like in this case, intersomatic vessel have no cherry expression, indicating that is post-transcriptionally repressed, but in the microRNA knockout, which lack of expression of the microRNA was the repressed. Now, although this microRNA function here and we have a full knockout, as I mentioned to you, by analyzing specifically this trait, which is the growth of intersomatic vessel, as you can see right here, at different time point of development between 26 hours post-fertilization to 54, they form quite normally between the mutant and the wild type. Although, again, in the constant to measure trait development, we decide to engineer with this microRNA knockout a transgenic line that visualize essentially two important things. First of all, by expressing the nuclear BFP, we could count the number of cells within this vessel that, again, are called intersomatic vessel. And second of all, by engineering the a membrane GFP, we can also count the essentially number of filopodia and literally understand the shape and length of this filopodia cell. Now, if you look essentially in the wild type, we can already observe that the shape of endothelial cell within intersomatic vessel have a quite large variability. But what was interesting in the mutant knockout, although again they don't have any phenotype and they have mildly increased in the filopodia number, when we look at the variability of the shape of endothelial cell, this was the most striking and significant difference. So we could literally found in the same intersomatic vessel endothelial cells that have a large amount of filopodia, as well as endothelial cells that did not have filopodia whatsoever. And this is again an increase of standard deviation. So this is a phenotype that is called increase in phenotypic variation. Now, a question that you may have right now is why we should care, since the overall phenotype of this mutant is fairly normal. With the idea again that a gene is perturbing a phenotypic variance of a phenotype is linked with the response of the environment. So again, to really understand the importance of such phenotype, we decide to took this animal and do a stress sensitization assay by using the intersomatic vessel growth as a readout of the sensitization of this knockout. And we used pretty much anything pop in our mind, including the drug screening for pro and anti-angiogenic compound, as well as temperature and oxygen. And this is what we found. So in essence, when we treat the microRNA miR-139 knockout with two different pro-androgenetic drugs, such as a vincinion and a Notch inhibitor, what we found that the wild type at this particular dose was, so to speak, protected by this perturbation, but the knockout mutant have this extra branched with the intersomatic vessel, both with the vincinion as well as the Notch perturbation. Interesting in that, this knockout could also be sensitive to anti-angiogenic drugs, such as using an inhibitor for VEGF receptor 2. And again, the Walters is protected, but the absence of this microRNA enable this vessel to be stalling, so to lack in their proper growth. And finally, by testing the temperature change in oxygen, we could see again that the microRNA type to respond to this environmental change much more robustly than it would do a wild type, which is seem to be protected. So what we identify essentially is that no matter what type of stress we adopt for this microRNA knockout, they always respond to the environmental stresses, although they are genetically have this perturbation, but they appear normal, at least in laboratory conditions. So for this reason, we start really to think about that non-coding RNA control two aspects of a phenotype. I hope that the literature, the previous literature, our literature, of course, support that they control the construction of a trait. What does it mean, the construction? In a sense, if you think about blood vessel again, the length of a blood vessel, the number of endothelial cell. So by the microRNA knockout, you do see a phenotype. In this particular case, the trait is changing. But now we also understood that the microRNA control as well the, did I do something? Oh, good. Let me see. Control also the robustness of a phenotypic characteristic. And by robustness, I mean, again, the variability or the variance of the phenotype. In some cases, microRNA knockout don't have absolutely no phenotype, so the trait per se will look normal across the population individual. In some other, they have a slightly increase or decrease, but overall, the most statistical difference is in the variance. So this type of study, it was published last year by a very talented postdoc, Diana Casper, really make us think about how do you build a trait? How do you make a trait? And by trait, we really means a collective cell behavior that for the most part responds to a genetic cue and will behave very similar. But there is also outlier on this population. And these outliers seem to be the more...
I'm sorry to interrupt you. We cannot see the big screen. We can only see the presentation mode.
Oh, I'm sorry. How about that?
That's perfect. Thank you very much.
Where do you lose me? Sorry.
When you had the problem that you said, oh, something happened that was not good, and you tried to restart it again, then it didn't restart completely.
Okay. Okay. Can I start from here?
Yeah, I think you can. Thank you.
Oh, yeah. Sorry about that.
No problem.
Yeah. So the way traits are forming, it really makes us think about what are the mechanisms by which these traits are essentially arising, and particularly thinking about the concept of a distribution curve in which the majority of the cell responds to a genetic cue and it will form in a certain specific way. And then, of course, there will be outlier, which seem to be particularly interesting on the point of view of disease susceptibility. So what could it be, the molecular mechanisms that drive this type of formation? Again, we discovered the microRNA knockout have this type of phenotype, but we really had no clue at the time what could be potentially the molecular target and so forth. So for this reason, we postulate an hypothesis that the trait or the distribution of the trait is controlled by an homeostatic mechanism. And homeostasis is a very fascinating mechanism, and I believe each of us have a little bit of interpretation of what is an homeostatic mechanism. So I give you what we believe at the time it could be an homeostatic system. So in essence, for us, it would potentially be the interaction between positive and negative regulator of that particular gene signaling pathway that control the formation of a trait. Why do we think it is a positive or negative regulator? Fundamentally because we could perturb the animal by responding in the positive way. If you remember, we can have more philopodium and more endothelial cell growth if we treat the animal with an androgenetic drug. But we can also flip the phenotype by having essentially the opposite phenotype if we drive the environment to be less androgenetic and so forth. So that's why fundamentally we postulate demonstrate the presence of powerful homeostatic mechanisms that control the formation of a phenotype. To answer the question of how microRNA can control this, we really have to go to the core of what microRNA do, and of course how microRNA function. And this is just a reminder, but it's of course very well known that microRNA are able to regulate post-transcriptionally their specific target gene by a Watson-Crick interaction of only a few nucleotides, which is called seed region, with the microRNA responsive element in the 3UTR. And this interaction is predominantly needed to have an RNA binding protein that's called Argonaute, which facilitate the binding and allow essentially microRNA to post-transcriptionally repress translation and also destabilize the mRNA. So the trick that we thought was important to do to understand what microRNA and microRNA network is present in endothelial cell is by performing a technique that's called AGO2 HITS-CLIP. So the AGO2 HITS-CLIP is essentially immunoprecipitation Argonaute on, in particular in our experimental setting, was done in both arterial endothelial cell and venous endothelial cell. These cells were first shined with a UV254, which clip Argonaute, and essentially this clipping or this cross-link is so powerful that you can literally treat the entire sample with the RNases, and therefore upon immunoprecipitation, upon digestion of the protein, you can capture the RNA by p32 labeling. It is only inside the core Argonaute, and then by doing Illumina sequencing, you can easily capture the species inside Argonaute, which presumably they correspond to a 3UTR mRNA-mRNA. And this is what we found. If you observe in a map, you use Genome browser, how these sequences look like, you can appreciate that they're predominantly presence in the 3UTR of genes, and interesting enough, there were no really difference about microRNA-mRNA network between artery and vein. And this peak are quite sharp, about 30 to 70 nucleotides. So they really indicate very precisely the footprint of where a microRNA and Argonaute are binding in the 3UTR. Now, once we had this data, of course, the first thing we decide to analyze if there were a particular signaling pathway in which this microRNA Argonaute complex in this endothelial cell, which presumably they are in a steady state, so to speak, homeostatic condition. And this is where we found a little bit of surprises. So what we actually identify is that 60% of the entire messenger mRNA in the Argonaute complex in this cell type, endothelial cell, are genes that correspond to cytoskeleton-adhesion-matrix genes, which is essentially I will call for now on a CAM gene. So this is a gene. They are very important, of course, for cell remodeling, cell adhesion, extracellular matrix. And I have this kind of busy scheme here, but this is actually the feeling that we have almost overwhelming enrichment for these actin and adhesion matrix gene in the Argonaute peaks. So we can literally find many components of the extracellular matrix, many components of the focal adhesion sensing mechanism, many other components of actin and tubulin binding protein, and finally some of downstream signaling of this mechanism. Now again, thalate integrin, which I'm assuming some of you may know, they are indeed the sensing mechanism of how cell senses extracellular matrix were not inside the Argonaute complex, and maybe we can discuss later about that. So the other thing we can find, of course, once you know the microRNA, the mRNA inside the Argonaute complex, associate then with the correspective microRNA. Well, it was a little bit, again, surprising to find that the majority of these CAM gene, in which they have multiple binding site per microRNA, are not really binded by one specific microRNA, but they are binded by many, and the level of redundancy, it really suggested to us the robustness of the mechanism. So in essence, if one microRNA is missing, there is 5 more that can bind the same gene, such as thrombospondin, serpin, etc., etc. So what is this, right? So in essence, if there is a strong bias of microRNA in a steady state for this CAM gene, we felt like perhaps what they're doing for living in this particular condition is regulating some fundamental mechanism. And really we have to go back to the idea of why these genes are so important, which is essentially for the Mechanotransduction of the matrix stiffness. And I want to start acknowledging by showing this slide that indeed the conceptual thinking of what's going on with all the genes was realized with my colleague at Yale and collaborator Martin Schwartz. So Martin at the time was actually write a very nice review, the Humphrey, and essentially what they were articulating this review, that's essentially how cells sense the mechanotransduction of the stiffness. So first of all, any type of extracellular matrix turnover or fourth generation is sensed by a cell via this integrin actomyosin dependent signaling pathway, which surprisingly, the cell, by sensing a stiff environment, for example, deform the extracellular matrix in gene expression by generating what is defined as a positive feedback loop. So the more stiff is environment, the more stiff is the cell. This has happened very nicely also to endothelial cell. If you take an endothelial cell such as UVAC and you plate it in a soft environment such as 3 kilopascal, or in a more rigid environment such as 30 kilopascal, you can actually see how the cell in a rigid environment is much bigger as far as stress fibers, F-actin staining, have many more focal adhesion, and does also have a much higher activation of YAP by nuclear translocation. And again, YAP is one of the major transcription factors that translate the mechanotransduction of the stiffness. Now, I think you pretty much all know, especially if you work in cell culture, that by putting a cell in a dish, plastic, which is high stiff, cells don't become bigger and bigger and bigger, perpetually at least. So they reach somehow a homeostatic shape. And that's why most of this biology as well as bioengineering, they know that there must be negative feedback loop that counteract this positive actin-myosin dependent contraction in a way that the cell reach their favorite shape, and therefore they try to maintain this shape. And this is a concept that is defined as Stiffness homeostasis. Now, perhaps when we have this data after the Argonaut HITS-CLIP, we start to think about that the number and the magnitude of the microRNA binding this active cytoskeleton gene, it could really represent the negative feedback loop that allows cell to reach this stiffness homeostasis. Of course, the challenge in this project at this point, it was how can we possibly validate and work with so many interactions, so many microRNA-mRNA interaction. And it was very fortunate at the time that a new technique was recently published about named sensor-sex library, which will really allow high throughput validation of microRNA and mRNA interaction. It's based to this bidirectional microRNA biosensor that I explained in the beginning with the miR-139. In this particular situation, again, the bidirectional promoters drive the GFP, and the cherry have a multi-cloning site in the 3UTR. So for us, it was very useful to take every single microRNA responsive element that we isolate from the Argonaut HITS-CLIP and clone each of them, again, 30 to 70 nucleotide in this lentivirus system. Just to remind you, by infecting a system like that in a cell, you can find cells that have equal expression of GFP and cherry. And in this case, there is essentially no regulation of the microRNA responsive element, but you can also find a situation in which GFP is equally expressed, but cherry is suppressed. And that will tell you which lentivirus driving the microRNA responsive element that is bound by a microRNA. So by having this in mind, we generate a library of CAM 3UTR comprised of about 100 of these microRNA responsive LTUTR in this gene. And this library was used to infect cell in two different substrates, again, a soft substrate and a more rigid substrate, 30 kilopascal. And the idea was essentially to understand in which of these conditions these microRNA are engaged age, and they're able to translationally block the cherry. And also, in addition to the sensor-sec, we perform also proteomics and RNA sequencing, as well as microRNA sequencing, which essentially give a readout of the protein output, essentially. Now, I just want to clarify how this cell gets separated depending on which receive the microRNA active or not. And it's basically quite simply actually via flow cytometry, in which we gate the flow cytometry with empty sensor with cherry GFP are equal, or with a positive control in which, as you can see, the GFP is highly expressed, but the cherry is highly repressed. And of course, we decide to run our library with this CAM sensor in a soft and a rigid substrate, and each single cell were separated in genotype, and this is just some control validation. Now, I'm going to show you a quite complicated circus plot that I break down in a multiple section to actually discuss with you more carefully to our result. But this was essentially the only way to pile up all these proteomics, RNA sequences, and source sequencing, macroRNA sequencing, et cetera, et cetera. All right, so let's start with that. So as you can see here in the half of the circus plot, you see different color which correspond to the completely suppressed, strongly suppressed lentivirus that was isolated in different cell. And what you can immediately see here, the cell that were seeded in a very rich substrate, 30 kilopascal, have way more microRNA sensor that were strongly repressed compared, for example, cell that were actually seeded in a soft substrate. So that already suggested that whenever you push a cell to a rigid environment, microRNA preferentially are binding and post-transcriptionally repressing this by using this microRNA responsive element. The second thing that we look at the microRNA expression in this cell, and it was nice to see that there was indeed an increased amount of microRNA that are differentially expressed when the cells are seeded in a rigid substrate versus in a soft substrate. Now, it is interesting now to analyze what happened to the mRNA of this CAM gene and the protein of this CAM gene. And with this inner circle right here in red, you can see the mRNA stability or the mRNA differentially expressed in these two situations. You can see somehow that it was an unexpected result. So the majority of the messenger mRNA of this CAM gene were somehow stabilized or equally expressed. And the protein output right here in green, again, here we are representing the differential gene expression of this protein, were either in change or increase. Interesting, when you put the cell in a soft substrate, the stability of the gene as well as the stability of the protein seem to be coincide with the presence of inhibition of the microRNA. While in essence, in this particular case at 30 kilopascal, although microRNA are indeed able to bind their microRNA responsive element, the overall transcription of the gene and the overall output of the protein is unchanged. Well, we believe that this is actually the molecular picture of how an homeostatic method should look like. So upon chain extracellular matrix, such as again increase on the rigidity of the matrix, there is an activation of positive feedback loop, which is via predominantly transcription. That's very well characterized, for example, by up translocation. But in the same time, we believe that the overall protein output is controlled by the negative feed loop to allow cell to reach an homeostatic shape. Now, if this is conceptual and more global idea to think about cell reaching their shape based on the matrix occur, we really had to prove more functionally that what are the effect by losing this regulation. The more logical way to think about that is by removing microRNA, we increase perpetually this positive feedback loop. So the first experiment that we did, we removed the effect of argonaute, which, I remind you, is a protein that coupled the microRNA with their TUTR. And we built this UVEC lacking of partially the expression of argonaute. And what we found out that the cell, even in soft substrate, they start to have a much bigger cell area and stress fiber, more focal adhesion, and very significant increase of YAP nuclear localization, as well as an increase of traction force, which is essentially the readout of the contractility of a cell. So again, indicating that there is a deregulation or an inhibitory pathway. It's also interesting to me to think about that this perhaps is not really a dogma for endothelial cell alone. And indeed, when we tried this experiment in human dermal fibroblasts, we observe a very similar behavior. Again, the fibroblasts were, with all the assay, much more stiff than the wild-type control. Now, I can see how people criticize perhaps this experiment because we are literally deregulating one of the major proteins that made the microRNA function. So we decided to do a several experiments, including eliminate, for example, DROSHA, which is also another important protein in the microRNA machinery. And we did observe that endothelial cell has, again, this increase of hyper-contractility and hyper-adhesive phenotype. But we also try to do something more individually defined, for example, by knock out the microRNA responsive element in each CAM MRE-UTR gene. And this is something that can achieve by CRISPR-Cas9, in which the guide RNA is designed on the microRNA responsive element in a way that upon the transcription of this gene, so essentially in the mutant case, the microRNA responsive element is gone, and the microRNA is unable to recognize specifically the regulation in this particular gene. And this is how this library were further validated. So again, there is a mild repression of each single protein that was targeted in this way. So what we identified, we did about a dozen of these CAM MRE-UTR knockout, and we found out that by assaying the cell area, YAP nuclear localization, total force, is that each individual knockout somehow were hyperactive in terms of cell area, YAP, and total force, although only a few of them, including CTGF, probably has a similar behavior to the absence of Argonaute, although Argonaute knockout is by far the most effective, is the most effective mutant to deliver these positive feedback loop. And this is not surprising. It suggests that there is really the presence of a network of the interaction rather than one particular interaction more important than other. Okay, so this is cell culture, what's happening at the tissue level. Now, as far as tissue level, we decide to use this three-dimensional culture assay by using, in this case, mouse fibroblasts. And the mouse fibroblasts were pinned in a fibrin gel with specific suture right here. So this is the tissue layer right here. Over time, particularly over 7 days, what happened that the cell will start to secrete extracellular matrix, and they will start their contraction. And of course, because they're suture, they will generate a particular tension. Now, we measure this tension in the microRNA absence model again by using Argonaute knockout mouse fibroblasts. And what we identify is that, again, by performing a cross-section right here, you can see that in absence of microRNA, the construct was way much more contractile, as you can see by the phosphomyosin, and also as you can see by the diameter of this type of assay 3D system. Although we measure overall the number of cells and that were not affected. So absence of microRNA increased the contractility of a tissue and as is a 3D model. But the overall reaching question, what happened to the in vivo system? Is this a mechanism that is present in our homeostatic condition in tissue? Well, to answer the question, we went through another model, which it is my favorite model ever, which is zebrafish. And particularly, why do we think about this particular model interesting? Because we thought about the thin-fold regeneration system as a proper system to measure the important mechanical homeostasis. I'll tell you why. So in essence, this is a very simple layer of tissue. There is a couple of epidermal layer with a structure that is predominant an ancient formal collagen and some mesenchymal cell. So this is a very thin and well-developed layer in a fish, and it's very interesting from the point of view of regeneration and wound healing. Indeed, if you take this appendix and you wound it, you at the first thing, after only 1 hour post-amputation, or even 30-minute post-amputation, a very profound contractile reaction. And after this contractile phenotype, you of course, as any other type of wound healing in mammalian as well, we have inflammation and cell proliferation and finally growth to repair this tissue. So perhaps we felt like this could be a very interesting model to assay whether the presence of the absent microRNA are challenging predominantly the contraction of a tissue, the mechanotransduction of tissue versus other type of responses. The first thing we use is a model that is a maternal zygotic argonauts knockout. And again, don't be fooled by the nomenclature. It's actually an hypomorphic model. It's not a completely knockout because it lacks of a 50 base pair at the C-terminal domain of argonaut. So it's a catalytic inactive argonaut. And as you can see by the measurement of macroRNA in this model, the macroRNA are, as you can see, diminished, but they're not completely gone. Again, the absence of microRNA will not allow the survival of the animal. So we found out the first important information that the morphology of this fin fold is quite similar between wild type and argonaut. But if you do use atomic force microscopy, which is a technique to measure the rigidity of a tissue, we found out surprisingly that the argonaut knockout have about 20 to 30% more rigidity in their tissue models. And this is a phenotype specific of the level of argonaut because I can reintroduce a human version of a wild type argonaut and rescue so to speak, the softness of this tissue. Well, not surprisingly, although these animals seem quite normal, they don't respond very well to wound healing. Indeed, the argonaut knockout mutant is way more slow in repairing the wound, and the repair of the wound can be again rescued by reintroducing argonaut. Interesting, we proved this for argonaut, but we also felt that we had to do some more specific manipulation, again, with one specific of this CAM gene. And it popped in our mind to use connective tissue growth factor because, as I showed you before, it was between one of the most regulated macroRNA, which inhibition control multiple assay, YAP, and stress fiber, and so forth. So the idea is that wound healing would upregulate both transcription and post-transcription regulation. We prove this by tricking the system. So we make a transgenic line, and we drive the GFP with the 3UTR that contain macroRNA responsive element for this CTGF-A gene. So this is the fin fold upon wound healing, about 0.5 hour post-amputation, and this is 2 hour post-amputation. And we measure essentially the intensity of the GFP. You can immediately see right here, there is a huge, a couple of fold change in repression of the GFP to UTR. So macroRNA are jumping and repressing this to UTR. But at the same time, if we check transcription of the same genes, CTGF-A, by coupling the promoter region with the GFP, and then by analyzing the wound at 0.52, it was actually very interesting to note that the transcription spike as soon as we wounded. So again, post-transcription and transcription were activated during the early mechanical response of the tissue. Finally, we analyze the cellularity of this mutant and understanding is really mechanical the only problem with them. And it is because we measure several pathways, several signaling, and also several phenotype, including proliferation with PCNA, which is, of course, is one of the major drivers of fin fold regeneration. But there were no different in wild type versus organelle knockout or the CTGF macroRNA mutant. Again, this is a mutant that lacks the macroRNA responsive element of one gene, which is the CTGF, as I mentioned to you before. And as well as apoptosis, it doesn't seem to be perturbed in absence of this hypomorphic mutant of CTGF. When we look at the mechanical property of the tissue, we actually saw the real phenotype. So again, to mention, the fibronectin and phosphomyelin YAP were the three type of assay in which we decide to check for mechanical response. As soon as you wound, you have a provisorial secretion of fibronectin, as well as actomyosin contraction, as well as YAP nuclear localization. But after only 2 hours, this mechanical response had to be released to allow, as I mentioned before, growth and inflammation to occur. Well, in both models, Argonaut knockout as well as CTGF microRNA knockout, this response was specifically altered. So the Argonaut knockout with the most effect have an increase of fibronectin deposition and a perpetual fibronectin deposition even after 2 hours post-amputation, which is coupled with increase of phosphomyosin activation and as well as prolonged phosphomyosin activation, and YAP respond very similarly. Now, in the case of CTGF mutant, they don't have exactly the same phenotype of MZ Argonaut, but they do have one of them particularly very representative, which is have a hyper-contractility 2 hours post-amputation, suggesting that the contractility or overall the mechanical property of this tissue is not driven by one single interaction, and this is just a quantification, but is driven by the presence of a network of post-transcriptional regulator. So with that, I would like to highlight, and I hope the main conclusion of this paper that indeed cell tissue at the tissue level or individually, they always have to counteract the effect of the matrix. This is because matrix do change in vivo as well in vitro by ACM2 mover or any type of fourth generation the cell may counteract during the lifetime. And how does it work? It works by transcription and controlling this very important gene that I mentioned to you, most likely via YAP nuclear localization, which trigger both the increase of CAM protein expression, but at the same time trigger a response from the non-coding RNA that couple the negative feedback loop and allow this protein output to be precise over time and over the response of the matrix. And I like the idea that matrix is the first environment that cell had to counteract, think about, and change, and that's how microRNA are potentially very good in regulating specifically this signaling pathway. So I thank already Tristan and Alberto, and I just want to give you an update of my current member in the lab. I talked briefly about Diona, and I thank everybody, and of course my collaborator at Yale, particularly Marty Schwartz and Tristan, that are working currently for him. And I guess now I'll be happy to take any question.
Okay, thank you so much, Dr. Nicoli. At this point, I'm going to turn this over to Dr. Driscoll to see if there are any questions from the audience to ask to Dr. Nicoli.
All right, yeah, we have a couple of questions here. So I'll just start reading through these. You can answer them one at a time. So the first question, is this regulation, this negative regulation of CAM genes specific to stiffness? Do you think that other forms of mechanical stimuli, such as fluid shear stress or stretch, might also be buffered in a similar way?
Yeah, I think it's, I love this question because it really essentially point out somehow how do we start from a vascular endothelial cell, and when we in vivo we had to couple more to an assay that is related to fibroblast stretch and so forth. So I think endothelial cell do respond to extracellular matrix, but in vivo they also have to respond to hemodynamics. In essence, how these two are coupled in an endothelial cell in vivo, I still don't know. I would love actually to study that. First of all, do we even know that hemodynamics is in a homeostatic setting? We know based on what we found that matrix is in an homeostatic signaling pathway, but again, we don't know nothing about how fluid shear stress may be in controlling similar ways. So that will be the first thing to answer. And I can tell that the Argonaut knockout does have vascular problem, including diminution of the dorsal aorta diameter. Although whether this is again purely the effect of matrix or hemodynamics had to be investigated.
All right, next question, sort of related to that comment you just made, but you show this in the context of wound healing. Do you have plans to look at this in other contexts, possibly cardiovascular disease or fibrosis in vivo?
Right. So I'm actually, I love to hear more about this because I think potentially what we are looking here is a protective mechanism of homeostasis that perhaps once we are in a fibrotic condition, the mechanism per se either is completely lost or nothing to do with that. So I envision this almost as an anti-fibrotic mechanism that allows cells to respond to the stiffness of the matrix without becoming hyper-contractile almost immediately. But I'm thinking about in a disease context when there is inflammation and a ton of chemokine, etc., perhaps this network fell apart, and at that point the cell becomes something else and transform it. And maybe we can think about that particular situation. One individual microRNA may function more than other. I mean, we know a lot of things about miR-29 in fibrosis and so forth. So the idea of microRNA individual function versus a network, I think the network is more important to preserve the homeostasis. So I will like to study this mechanism in a more physiological context rather than in a disease setting, although I do believe that this interaction between CAM gene and microRNA could be maybe a marker for early fibrotic event if it's real that they are the first anti-fibrotic type of response.
All right, then one last question here is, do you think that this system of microRNAs is similar in all cell types? You show it for a few cell types. Are there similar microRNA in these cell types, or is it unique to each type of cell?
Right, so as far as the microRNA that bind the CAM gene, we've done an analysis of the microRNA expression between fibroblasts and endothelial cell when they were seeded in a stiff substrate, and they are actually quite similar. So again, the amount, the magnitude in terms of level of expression and the type of microRNA between endothelial cell and fibroblasts in stiff condition are very similar. So suggesting that indeed there is this global response. Although again, we analyze predominantly human cell. Now, if you think about other cell type from other species such as mouse or even zebrafish, we can also speculate that different microRNA could be used because the two TR sequences are slightly different between species. So therefore, new interaction, they may be present and regulated. But within a species, I really think that there is this common repertoire of microRNA, and I think you really want as many as possible to guarantee the robustness of this mechanism, due to how important it is to keep the cell shape as well as the matrix in check throughout the life of a cell.
All right, that's all of the questions that were entered in, but if anybody wants to raise their hand and ask more, you could do that now.
Do we have any hands? Anyone? No? Well, if you're just shy and you want to pose your question later, you can email info@navbo.org and we'll make sure that we share your question with Dr. Nicoli. At this point, I hope you found this seminar beneficial. These webinars are brought to you by the NAVBO Education Committee. Our next webinar is March 14th, and it would be presented by Strider Meadows from Tulane University on angiopoietin-2 inhibition, rescues arteriovenous malformation in a SMAD4 hereditary hemorrhagic telangiectasia mouse model. Look for the registration information in the NAVBO newsfeed. Also, please fill out the evaluation form at the end of the webinar or when you receive it in a subsequent email, and let us know what you thought of today's webinar, and importantly, any topics you may want to see in a future NAVBO webinar. This helps us a lot with organizing the NAVBO webinars. Thank you very much. Thank you, Dr. Nicoli. Thank you, Dr. Driscoll.